So our next speaker is Andreas Meyer from Fgen in Basel, Switzerland. And yeah, thanks much for that talk again. And we all know that, yeah, in synthetic biology, we can build a lot of stuff. We can build a lot of stuff rapidly and at large scales. But I guess one of the biggest challenges is also the screening. And I think Andy has a very powerful and interesting technology developed with his company that facilitates a large number of screening, in particular microbial strains. Andy is from Fgen in Basel, and Fgen is a spin-off company from the Swiss Federal Institute of Technology from the ETH in Zurich, and they are particularly interested in microbial strain development for different industrial customers. As an entrepreneur, Andy is particularly interested in technology-driven companies and how open innovation can boost product development. Andy himself also did his PhD at the ETH in Zurich and on the work of engineering proteins for biocatalytic applications. Welcome, Andy, and happy to see you, even though it's virtual.
Hey, thank you, Dominic, for the nice introduction. I hope you can give me a thumbs up if you can see the screen nicely. Okay, perfect. Okay, I'm happy to present our company and how we believe that we can accelerate the SynBio cycle by improving the testing. As we have heard before, we need a lot of data to learn, and we think we have quite a unique technology that enables us to get this data. So first, one word about our company. So what is Fgen? You probably have not heard of us before, although we are already like 10 years active. So we spin off from ETH Zurich in 2011. We are located in Basel. And what we do design and optimization of cells and microbes. And we do this together with the industrial partners. So we really work on real-life problems, so we improve strains for our partners that are afterwards used in bioproduction. Here also, of course, you all are familiar with the design, build, test, and learn cycle, so with the traditional SynBio cycle, and we have also learned what different methodologies we have for all the different steps. So there have been a lot of progress in the modeling in the recent years. We know that DNA synthesis is really a nice tool that we have now available, which was not here 10 years ago to build. In the testing, we have automation. As we have heard, there is foundries that do automation. We have DNA sequencing that also got very efficient. And in the learning part, we have the artificial intelligence and the machine learning. So from a commercial point of view, we see that this is like or the distribution of this work is like that. We have a lot of tools available for 3 of the 4 parts, but the testing still costs a lot of time and thus also from a commercial perspective, a lot of money. Why is this so? So let's have a little example and look at the very simple pathway for metabolic engineering. So we have like 3 open reading frames that we want to have in the pathway, and we just have like 5 different promoters and 5 different ribozymes that we want to randomly combine with these 3 genes. So we end up with approximately 100,000 combinations, okay? So when we do this in typical Erlenmeyer flasks with a throughput that we say now 1,000 per week, we are 7.5 years doing the testing of all these combinations. If we do it in microtiterplate, it already looks better. If we work 24/7 and need 1 minute per assay, we can do 10,000 per week. So it takes us a little bit more than half a year. So if we use our nanoliter-reactor technology, we are finished by lunch, okay? So it's a little bit another throughput that we have here. And how does this work? I would like to present you today. So what is the nanoliter-reactor screening platform, as we call it? So we start with large cell libraries, typically in the 100,000 to million range. We use cell encapsulation, and we use cultivation in these nanoliter reactors to cell clusters. And Afterwards, when we have these cell clusters, either during cultivation or after cultivation, we apply a phenotypic assay for the specific property that we look at. And we use a flow cytometer for analysis. So this enables us to analyze at very high frequencies. So I say at 100 events per second, we can easily analyze. Afterwards, when we have isolated or analyzed these nanoliter reactors based on their fluorescence, we can isolate the strains out of the nanoliter reactors. We characterize them in a secondary assay. And if everything works fine, we have the cell with the desired properties. And the whole workflow or the whole technology is characterized on one hand by the throughput, which is orders of magnitudes higher than competing technology, but also the sensitivity and the robustness are very important characteristics. So if you look at different organisms grown in nanoliter reactors, so these are some microscopic pictures of, you see a bacterium, E. coli on the top, in the middle you see a yeast, so Yarovia lipolytica, and at the bottom you see filamentous fungi where we encapsulate spores, and which also then you see the filamentous growth. If you compare growth and productivity, they are exactly the same as in liquid culture. So it really represents also the situation during the screening that you have later on when you apply your microorganisms in the process. The key, of course, is the fluorescent readout. There is different assay formats that we apply. So very simple assays would be proliferation assays, where you just look at the size of the microcolony. You can use intracellular switches and do intracellular assays. But since most biotech products are secreted, the secretion assays are the most important or most often applied ones. And there we have two basic principles. So one is functionalized nanoliter reactors, where the compound of interest or the product or the target that is secreted from the cell triggers a fluorescent signal in the nanoliter reactor itself. Or the other one is the co-cultivation, where we co-cultivate together with a candidate strain, a multiplicity of sensor strains, and these sensor strains then react on compounds secreted. So very simple example, you have an auxotrophic strain for amino acid outside, your candidate produces an amino acid, the biomass formed outside directly correlates to the amount of amino acid that is produced. I want to go to two specific examples that you can get a little bit a better understanding from this abstract description, how we really use it in projects. And I selected here a recent example from Valentina, which is a PhD student in our labs, which really, I think, is a nice example of the whole SynBio cycle, okay? So the goal of the project is to increase the protein secretion, in this case of an amylase in Bacillus, okay? So the design of the library was based that we looked at all the wild type signal sequences for protein secretion and defined parameters which we consider as important. These are 267 parameters. It's like charge, hydrophobicity, RNA structure. So we take into account not only the amino acid level, but also the DNA level. So we ended up with a library with about 11,500 sequences that we designed in silico. We built it then, or we synthesize the library by Twist. So since it's a sponsor today, I'd like to mention Twist. And integrated the sequence into the genome of the strains. We were successful in about building 90% of the strains that we have targeted. So we built up about 10,000 strains. And in the testing, we wanted to have a multiplicity of testing of at least 10. So what we analyzed in the end, we analyzed 160,000 samples. So that means we had an oversampling of about 15. And we detected about, again, 90% of the strains or of the sequences in the end. So about 10,000 of the 11,500 sequences that we targeted were, let's call it biologically viable and leaded to really a phenotypic assay. And then we used this data, so we isolated the samples, we used DNA sequencing for identifying the sequence of the whole signal peptide, and then used machine learning and prediction to define design rules for signal peptides for the secretion of these amylases, okay? And in the lab, it looked this like. So this was the assay that we have developed. The assay principle is actually quite simple. So amylases degrade starch, okay? So the assay principle is that we loaded the nanoliter reactor with fluorescently labeled starch, okay? If now the cell proliferates and secretes the amylase, the starch is degraded and the fluorescence of the nanoliter reactors is reduced. So the fluorescence of the nanoliter reactor inversely correlates with the activity of the amylase or with the secretion of the amylase. You can see here on top a very homogeneous labeling of the nanoliter reactors with the fluorescently labeled starch. And if you now introduce an amylase secreting strain, you can see it in the picture here. We use a cell staining for the biomass, and in this case it was a red. You see here the colony grown, and you can see the reduced fluorescence of the nanoliter reactor in comparison to a nanoliter reactor that does not harbor a colony and is highly fluorescent. So in the experimental testing, we used then these two values, so the fluorescence of the biomass and the fluorescence of the enzymatic activity in our selection procedure. So you can see here it's substrate degradation. So the green fluorescence on the y-axis indicates the amylase activity, and the red fluorescence, the colony size. So we defined 10 different bins based on specific activity. We collected all the strains in specific bins in 10 different vials. We sent them to sequences and had them the sequences available and compared the sequences. What is the sequence of the signal peptide that makes a very good or a very bad secretor, okay? And using machine learning tools and prediction tools, we came up then with design rules. So from the originally 267 parameters, about 150 parameters have turned out to be relevant. And what you can do then, you can look at the individual parameters in such a way. So in a very simple spoken, you see each value. So in red is the corresponding feature as a high value or a low value. And everything that is here to the right decreases secretion, and everything that is to the left increases secretion, okay? So I just want to pick examples. So the first two are probably the most easy to understand. So if you look at the total hydrophobicity of the signal peptide, the design rule will look like if you have a low hydrophobicity overall, the secretion gets bad by tendency. If we increase the hydrophobicity, secretion is better. Or a very yes or no answer is here. If an alanine is in position -1, the secretion is good. If the alanine is not there, secretion is bad. And then if you take all these different parameters for the prediction, you can actually find out and learn why a signal peptide is bad or why a signal peptide is good. So we confirmed the model that we had by taking a number of bad secretors, you can see them here, and good secretors, okay? And what you can see is the original sequence in white. So this is the native activity. So one is the activity of the native signal peptide for this amylase in Bacillus subtilis. And what you can see, the bad secretors, of course, have less secretin, and if you edit, at least two of them almost can reach the level of the wild-type peptide. Okay, so we just took simple examples where we just changed one feature. Of course, it works very nicely if you say we make a good secretor bad, so you can apply the same design rule, and there it works, of course, much better because also it's much more difficult. But the really cool result is here. So these are then sequences which are really de novo designed. for the secretion of these amylase. And what you can see that approximately half of the sequences that we designed in silico were as good as the wild type. And even some of them were even better. And if you say now, okay, I have to design like 20 signal peptides from scratch and test them, that's really not a big effort. So I think this is a very nice example of the whole SynBio cycle for a relevant problem, which is the secretion of industrially relevant enzymes. The second example that I want to show a little bit in short is the co-cultivation assay. So for how works the co-cultivation, the principle is here that you have the candidate cells in the nanoliter-reactors, you have a multiplicity of sensor cells, and these sensor cells react to a compound that is secreted by the candidate. So in this assay, we were looking for antibiotic peptides. The library was designed such that we took a number of known antibiotic peptides or antimicrobial peptides and shuffled different domains of these peptides with each other, and designed also a library of approximately 6,000 variants in this way. Screening was done again in nanoliter-reactors. You can see here an example where a library clone, again in red, shows no activity, and where a library clone shows very high activity. As you can see, the biosensor strain that is co-encapsulated is completely inhibited in growth. And again, we isolated a large number of candidate strains and looked at the modular diversity and of the activity, and were able to isolate really, on one hand, to define design rules. So it was clear that at the N-terminal part, both modules that we selected work fine. Also, these are in the second domain. It's the only two modules that work fine, is really from these two antibiotics. And we see that the design rules, in the end, there is much more flexibility. And but what's more important, we really were able to isolate new antibiotic peptides with a changed substrate specificity or host specificity in this case. So if we want to close up, and the nanoliter reactor platform is really a platform that enables you to accelerate the SynBio cycle. It's applicable for any cell types that you like to work with. So we did project with bacteria, with yeast. You see here microalgae. So this is chlorella, which is a phototropic growth microalgae. You see filamentous fungi. We worked with mammalian cells. So you see here CHO cells, but we also work with cancer spheroids. And for a variety of products, proteins, chemicals, biologics, and cells. So here, really the key is to design an assay that represents the features that you're looking at. And if you compare it to the microtiter plate based systems, of course, we are orders of magnitudes higher in the throughput, but also the cultivation in these hydrogel beads or in this nanoliter reactor is much more close to the bioprocess conditions, or to the conditions that you afterwards apply your cells. And it gives you the ability to really generate a lot of data, make the design rules, and then start to rationally design biological systems. So, and what we do? Yeah, we do at Fgen, we use biological engineering and combine it with a screening platform for the design of new strains and new products. And of course, we are always open for collaboration. So also with academic partners, if you have cool projects where you believe that our screening platform can support you in your either data generation or strain design, please drop me a note. You will find the contact data here, and I hope you have now a lot of interesting questions. Thank you.
We've got quite a few questions for your talk. The first one from Bajek. have you run into any problems on the FACES-SEC step?
I have to say I was not doing the machine learning stuff. Okay, I mean the work is not done performed by me. It's better if I ask the expert how it's really done. So he can drop me a note, and I'm sure the person that has done it, the PhD student, is very happy to communicate further. But I would probably not be the right person to answer this question.
The next question from Leonardo is very fantastic talk, Andreas. How stable are these nanoliter bioreactors, and how do you supply oxygen?
So they are very stable. So they have some stuff that they don't like, but in principle, they are temperature stable, they are medium stable. They can be, you can do PCR in it. So you can heat them up to 95 degrees and you can put them in typically all cultivation media that we use. Oxygen is not a problem because we just take the nanoliter reactors and put them in a shaking flask and put them on the shaker. So since the cell density is much lower than in a normal shaking flask culture, so we start with typically 1 million cells, and if you inoculate the shaking flask, it's much, much more. So we don't have oxygen limitation or pH problems, so it's very convenient. It's not to compare with a droplet-based system where you have like an organic phase or an oil and a water droplet within the oil where you have oxygen limitation. It's not to compare with that.
So 1 million cells can fit inside each of, 1 million living cells can fit inside each nanoliter bioreactor?
No, of course we have just one candidate per nanoliter reactor, but we can put in 1 million or nanoliter reactors in the shaking flask.
Okay.
So therefore, the cultivation in the nanoliter the cell density during cultivation is very low.
Okay. Do you use random libraries, or do you employ some sort of intelligent design with these experiments?
So we do both. So we do also random libraries, but there's 2 reasons for that. If the obvious targets already have been engineered, we do just classical strain engineering by UV mutagenesis or chemical mutagenesis. And the other is if our partners want to keep the non-GMO label on the product. So that's very important. So we are here, the SynBio community, and don't understand, but still 80% of the consumers believe that GMO is detrimental to your health. So if you have to sell a product, you want to have the non-GMO label. And therefore, even being a synthetic biology company, we do a lot of random mutagenesis because there you really need the throughput, and there we come into play.
Okay, and one last question from Daniel. Is the technology compatible with fluorescent labeled antibodies, or is it limited to reactions that yield fluorescence?
No, you can do an in-bead. We call this the in-bead ELISA. So you can immobilize the protein inside and then take a detection antibody or secondary antibody and make an ELISA, actually a sandwich ELISA, where you immobilize the protein inside and use an antibody for detection of the protein.
Okay, fantastic.
I show examples of that because I was told it's very strictly limited to 20 minutes, and I thought I show the ones that are most close to the synthetic biology.
No, brilliant, brilliant. Thank you very much. Unfortunately, that's all the time for questions that we have. Thank you again for your brilliant talk.